Welcome back. Now we're going to talk about the process of transcription. So, just a refresher. The genome is all the genetic material of an organism. The human genome is about 30,000 genes long. Now, genes are not defined by a certain size. They're small segments of DNA that contain the information to construct a specific protein. So you can think of one gene, one protein. Now, in reality, we understand that it's a little more complex than that. We know that some genes are responsible for multiple proteins and multiple processes, depending on how they're read. But that is above where we need to be. So, for our purposes, our understanding is that one gene codes for one protein. So, you may have a gene that's relatively short. Say the gene to make the protein elastin. Now, elastin is responsible for letting your skin and tissue snap back to its original shape after being contracted. Kind of like when you poke your cheek, your skin bounces back. It's not like a piece of fruit where, like, if you stick your finger in it, it's dented and then it stays that way, right? Elastin helps your skin rebound. Now, there's another gene that makes a protein called factor VIII. And we'll talk more about factor VIII in topic 6. This is a protein that helps blood clots to form. And this is there is a mutation in this gene in people with hemophilia, so that protein is not properly created. So there's a mutation somewhere in here, if you're hemophiliac, that prevents the proper shape and formation of that protein. So we're taking the next step in talking about how this information in DNA is transmitted to the ribosome. Now remember, DNA hangs out in the nucleus, right? Transcription is the synthesis of mRNA copied from DNA by RNA polymerase. Now, that's a lot of words. We'll talk about what those words mean. But like I was saying, your DNA hangs out in the nucleus, all right? Eukaryotic cells store DNA in the nucleus. Your DNA is what contains the instructions on how to build those proteins, and those instructions, right, are your genes. So you can think of, if your DNA is an instruction booklet, a gene might be one page in the book or one paragraph in the book that tells you how to build a specific thing. But our proteins are built by ribosomes out here, outside of the nucleus. Here's the nucleus, here's the DNA, right? Here's the ribosome. This is the builder. So we need a way to get this information out. Now, the entire purpose of having a nucleus is to protect your DNA. So DNA is not allowed out. That's not a thing. It stays inside. So transcription is the process by which RNA is going to create a copy of DNA instructions that are allowed out of the nucleus and can travel to the ribosome. Now you might think, well, that's stupid. Why don't we just let our DNA out of the nucleus and then the ribosome can get there, it can get it sooner. So prokaryotes, that's how they work, right? So prokaryotes, they don't have a nucleus, remember? So their DNA just kind of floats around, and the process of translation and transcription happens simultaneously. The problem is they tend to have a lot less DNA, so that process is relatively simple. When you're talking about 30,000 genes, that process gets kind of complicated, and you don't necessarily want all that stuff getting mixed up. So our DNA is protected in the nucleus. And that's fine. That's what we have RNA for, so we're good. So let's review what RNA actually is and how it's different than DNA. So remember, DNA has 4 different base pairs: adenine, thymine, cytosine, and guanine, A, T, C, and G. RNA is similar. It still has a phosphate. It still has a sugar, but instead of deoxyribose, it's just ribose. So there's no oxygen missing. And we have different base pairs. So remember, DNA has 4 different base pairs: adenine, thymine, cytosine, and guanine, A, T, C, and G. RNA is similar. It still has a phosphate. It still has a sugar, but instead of deoxyribose, it's just ribose. So there's no oxygen missing. And we have different base pairs. So phosphates, the same. Sugar, similar, but DNA sugar, deoxy, that's missing an oxygen. Deoxy minus oxygen. RNA isn't missing anything, so it's complete. And then we have the difference in the base pair, right? So we have uracil instead of thymine. Those are the major differences. There's also the fact that DNA is double-stranded and RNA is single-stranded. Okay, transcription is the process where we're going to create this RNA strand. This takes place inside the nucleus. And because of complementary base pairing, the information in this DNA molecule is copied, right? And we call this mRNA for messenger RNA. It's going to message. It carries a message. It carries the information in DNA to the ribosome. It's the messenger. All right, so let's see how this works. So, hey, check it out. We have RNA polymerase instead of DNA polymerase, because remember, we're not making DNA, we're making RNA. So, RNA polymerase, ACE enzyme, polymer, an enzyme that builds polymer, it's building RNA polymerase. So, during transcription, the nucleotides of a gene will be used to create a strand of RNA. This begins when an enzyme called RNA polymerase separates the double helix in the strands of the DNA gene. So no longer are we talking about helicase. We're talking about RNA polymerase. And that's important because RNA polymerase actually does more than one job. Not only does it separate the double helix, it also bonds the new nucleotides to the template strand or to the coding strand. All right, so I'm getting all mixed up. RNA polymerase does both. So RNA polymerase unzips the DNA and adds nucleotides. All right, so what do you think is going to go there? Remember, it's RNA, so there is no T. Instead, we use U. Can I select U? There we go. Nope, that's unfortunate. Okay. So we keep adding those nucleotides. And now we have our strand of mRNA. The RNA is going to separate. And we're going to say goodbye to our DNA. Okay, so we're not exactly done. This is pre-RNA. It has to go through a process called processing. RNA processing. So you'll notice that in the strand, there are some capital letters and there are some lowercase letters, right? And you're probably thinking, what the crap is up with those lowercase letters? Well, the lowercase letters represent nucleotides that are not involved in the making of a protein. Think of them as like extra letters, extra words in a paragraph that you don't need, right? So we're going to cut those out. We call them introns. Now, I know that's confusing because we're cutting them out and they're called introns, but there's a way to remember it, right? So these introns that we don't need for the gene, we're going to remove those. Everything else is called an exon. So there are processing proteins that come through and remove those introns and reconnect that mRNA strand. Now, the exons make up mature RNA. Exons exit the nucleus. Introns stay in the nucleus. So now this exon, this mature RNA, is going to leave the nucleus. So here is our nucleus. We have our double-stranded DNA. We're going to, through the process of transcription, make mRNA. Right? And that mRNA is going to exit the nucleus through a nuclear pore. And it's going to travel to the ribosome. That ribosome is going to use that information in mRNA to produce a protein. Did I spell that right? Yeah. This process is translation. Okay? So, step 1, transcription, right? Then our mRNA leaves the nucleus through a nuclear pore, travels to the ribosome, where the process of translation begins. And that's the actual building of the protein. So, just for practice, I would like for you to determine the RNA code that would be produced from the following sequences. So pause the video, and I want you to give it a shot. Now, I know you could just keep watching and just copy down the answers, but here's the thing. So I have seen questions on the exam that ask you to do this. And no one's grading you on this, so just give it a shot. You're allowed to screw up. Pause the video, try it yourself, and see if you can get it right. Okay, hopefully you paused it and gave it a shot, and let's see how you did. Hopefully you did pretty good. That was skill number 4, and that is the end of our lecture for today. But let's watch this.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
So, did you figure out that that's exactly what we were talking about? That is the process of transcription. Let's watch it one more time, and I'll turn the volume down and maybe narrate myself a little bit. So, one more time. Let's turn this volume off. Okay, so here we have our double strand of DNA. We have some processing going on here. It looks like we're going to get ready for the start of transcription. So this is a primer that's going to tell the RNA polymerase where to start. So the primer is looking for the sequence that tells it to start. Once it finds it, this is your RNA polymerase. Now remember, your RNA polymerase does two jobs at the same time. The RNA polymerase is speeding down the double helix of the DNA because remember, it does both jobs. It both splits the strand and forms the mRNA molecule, and it also reforms the bond or the DNA strands together. So it actually has three jobs, right? So it separates the strand, adds the RNA nucleotide, which is the yellow thing, the yellow strand coming out the back, and it reseals the DNA double helix. Three jobs. Here are the free nucleotides that are coming in that are going to be matched to make the RNA. And a lot of people ask, well, where do those come from? Those are just kind of floating around, hanging out in the nucleus. Those nucleotides. You get those nucleotides from the food that you eat. So DNA and RNA in the food that you eat, right, is broken all the way down to those nucleotides and then delivered to your cells. And they just kind of hang out in the nucleus. And they can be reused over and over again until they don't work anymore. So here we have that RNA polymerase. It's splitting the strand. You can see it splits back here. It's adding those nucleotides, and it's reforming those hydrogen bonds in the DNA. And that was it. That mRNA strand is then going to leave the nucleus, travel to a ribosome, and be used in transcription. But that's next rotation. So if you have any questions, you can respond to this lecture with questions in the comments. You can send me a message on Canvas. I can set up a discussion board. We'll also have a live lecture. Let's see, today's Tuesday, so we'll have live lecture on Thursday. And I'll see you then.